Hello everybody, this is a flip lecture for us to review some basic stuff about meiosis and also to look at some sources of genetic variation that we've been talking about in sexually reproducing organisms. The first thing I want to do is just go over some basic stuff about the inheritance of diversity and like what causes that. And what we do know is that DNA is deoxyribonucleic acid. It is stored in these nice packages that we call chromosomes. Okay. And these chromosomes, the DNA is actually stored in this part of the chromosome. This part of the chromosome is actually called a chromatid. So these are called chromatid. And this middle part that holds them together is called the centromere. And the centromere is pretty much a little protein disc that holds it together. When you reference each of these, they are referenced as sister chromatid. Okay, so we inherit these from our biological mother and biological father, and one chromatid contains DNA while the other one contains DNA as well. We learned that in order for us to inherit these genes that we find in the DNA in this, um, we have to have received a gamete that carries that information. And gametes are made through the process of meiosis. Okay, so meiosis starts out with a cell that we call 2N. 2N is called diploid. So in ova or in ovaries and in testes, there are cells that start out as 2N or diploid, meaning that they have a full set of chromosomes. In this case, with humans, we have 46. And throughout a man's life, as they reach sexual maturity, um, these diploid cells will become haploid cells. But in order to become haploid cells, it has to go through two divisions. And that's what meiosis is. Meiosis is two divisions. We call these reduction divisions because we're reducing the amount of genetic material in them. So here's meiosis 1, and you guys have this in your notes already. And here is meiosis 2. And during meiosis 2, then each one of these cells divides into two. Okay? So then what we end up with are haploid cells. And these haploid cells are just N. Okay? And they have 23 in each when meiosis is okay. When all the chromosomes separate properly, crossing over occurs, all of that stuff just works and it functions. Now we do know that there are some abnormalities where the chromosomes will fail to separate, or genes from one chromosome will jump to another chromosome, and those are different ways that we can get genetic variation. So let's move on to that. The first thing is crossing over. We need to know that crossing over occurs during prophase 1 of meiosis. So just to review this, you guys don't have to know the phases per se, but you do need to know some significant events that occur in meiosis. So I guess reviewing those phases would be good. It spells out PMAT, okay? So it's prophase, metaphase, anaphase, telophase. And I don't want to review that again because you guys learned it last year and the year before. But crossing over occurs during prophase 1. This happens when the chromosomes line up. So let's say this is from mom, and then this one is from dad. And then crossing over occurs when one chromatid here will cross over with this chromatid here. And what they'll do is they'll actually exchange genes. And the genes that they are exchanging, you guys, are actually the same type of genes. They're at the same locations. These locations are what we call the locus. So the location or the locus of the gene is the same. The information inside of it is different, okay? And that's what creates diversity, okay? Crossing over is essential to creating situations where it creates a mix-up of genetic material, because the too similar of genetic material can result in all kinds of complications, which we've already talked about. So that's crossing over. Again, this happens during prophase. And in meiosis, meiosis 1 starts out with interphase, but it's not technically a phase. And then it'll go through all these, and then it'll jump to PMAT again, but it is PMAT 2. That's meiosis 2. And the same thing here. Oh, this is 1. Okay, next thing, the law of independent assortment. I want you guys to think about the cell and the nucleus inside and all these chromosomes that are in there. What we need to know is that these chromosomes, think of it as this big dance party. And when they are called to line up and separate during like metaphase and anaphase, they line up independent of each other, meaning that they will assort themselves in a random way. So what I'm saying here is that, let's say um, these green ones that I'm circling are all of the maternal um, chromosomes, and then the red ones I'm circling are all paternal. So maternal means mom, paternal means dad. What I'm saying with the law of independent assortment, what this states is that not all the green ones are going to go to one side and not all the red ones are going to go to one side. They will actually randomly assort themselves, so they'll mix up. So that means that maybe a red one will go with 2 greens on this side. Maybe a red one, 2 red ones will go with 1 green on this side. This also, if you look back at the title, is a source of genetic variation because they're randomly lining up and moving around. Okay, so again, this is a source of genetic variation. This creates genetic variation. The next one is fertilization. This is spelled wrong. Fertilization, so we've got a little spermy dude, and then we also have an egg. The stuff inside of each of these is 23 chromosomes, 23 chromosomes. Those chromosomes are different, meaning that if we look at a karyotype, and this is site 1, if we look at site 2, and if we look at site 3, I'm not going to write them all out. The one that's from dad, let's make it blue, and then the other one is from mom. That means that at the locus of this particular chromosome right here, this chromosome here from mom that has the same gene. It's the same gene. So let's say that codes for hair texture. The mom, the maternal chromosome, has curly, and the paternal or father has straight hair. So that gene basically is going to have those 2 different traits there. And in the next unit, we're going to learn about the whole dominant recessive thing. But what I want you to know is that with fertilization, you're getting genes for the same trait that are different. In some cases, they will be the same, and that's fine, but it's all random, okay? So it's important to know that. So fertilization creates, go back to the title, genetic variation. It creates genetic variation because we're inheriting chromosomes that have different genes for different traits. Nondisjunction, this is the one that we most recently just investigated. This is when chromosomes, I'll put chromos for short, fail to separate properly, right? What we want are 23 chromosomes in our haploid cells. So here's a little sperm of 23. But during um, anaphase and metaphase, basically the chromosomes can fail to separate properly, specifically more anaphase. So if you remember, when the cell is about to divide and you have these chromosomes being pulled over from one side to another during anaphase to eventually make two cells in meiosis 1, these chromosomes, let's say that there are 6, these chromosomes will fail to separate properly. So let's say this side gets 3 and this side gets 3. And then during meiosis 2, anaphase 2, those chromosomes will pull apart again and create a situation where you have maybe 2 chromosomes or one, let's say 2 chromosomes more than another, okay? Or really one more. So what happens is these chromosomes fail to separate properly, okay? I'm going to provide you guys with another video clip of this, and I'll put it on our YouTube site for you guys to listen to and take notes on over the weekend to help prepare you for the quiz. So again, just know that chromosomes fail to separate properly, and what we end up getting are gametes that are carrying too many chromosomes, and then as a result, when that combines with another gamete, you have more than 46 chromosomes in the individual. And then mutations is just when there is a mix-up in the codons. If you remember, a codon is the 3-letter sequence. Okay, so I think our example in class was cat, and so that would code for GUA, and then we would look up the amino acid, and that would be fine. But let's say that the other one, the mutation, was CAG instead. That means that it's transcribed into GUC, and then you would look up what that amino acid is. If that amino acid is different, then it would create a different protein, and it would create a denatured protein, which can lead to a mutation. Okay, a denatured protein. Okay, and again, that's that with genetic code table, GCT, the genetic code table, where we look up these things. Okay, remember, sometimes mutations are helpful, sometimes they're harmful, and sometimes there's no effect at all. If it would code for the same amino acid, there's no effect at all. Okay, all right, that's the end of this lecture.